Hello, good morning. So I am Dr. Abdul Bari, assistant professor, Riyadh College. So today I am going to discuss about novel drug delivery systems. The scope of novel drug delivery systems. Novel drug delivery systems, one of the fastest growing healthcare sectors, with sales of the drugs incorporating novel drug delivery systems increasing an annual rate of 15%. According to the BCC research, the global market for the advanced drug delivery systems were valued nearly 176 billion US dollars in 2012 and is expected to increase to nearly 212 billion dollars by 2018. There are great opportunities for the companies investing in R&D for new improved drug delivery systems, allowing for improved therapeutic absorption and efficacy in the patients. Global drug delivery market by administration mode, so by the route of administration. So what is the global drug delivery market? So if you see oral drug delivery market, it is having 53% share. If you take inhalations, that is 32%, transdermal systems 8%, and injectables and implant 3% market share, and ocular systems and nasal systems 2%. Why novel drug delivery systems? Why we need to develop novel drug delivery systems? Because to optimize drug therapeutic effect and convenience, to enhance a product life cycle, to improve patient compliance, to target the drug to the various organs, to control overall healthcare cost, to facilitate biological drug delivery. The NDDS industry comprises of companies seeking to develop. So the companies are looking for novel alternatives to existing delivery systems. Example, implantable pumps. There is new techniques. They are implemented by the companies, and enhancement of existing systems. Example, so instead of conventional drug delivery systems, so we are using sustained release oral dose or oral dosage form to reduce the dosing frequency. Drug targeting to the particular organs or tissue, so like nanoparticles and viscular systems. The history of the NDDS. Okay. The novel drug delivery companies have existed since the late 1960s when Alza and Elan, so these are the two brands, pioneered the oral method of enhancement of the drug delivery. So that is example is ORAS, osmotic control release oral delivery. The types of the novel drug delivery systems. So if you see this one, so first one is very important, viscular systems like liposomes, nanoemulsions, neosomes, lithosomes, and solid lipid nanoparticles. And nanoparticles, gold nanoparticles, quantum dots, nanoshells. Transdermal systems, transdermal films. Organ targeting. So the targeting of the drugs to the brain, cancerous cells, colon targeting, eye and lymphatic tissue. And microparticulate systems, that is microspheres, pellets and mini tablets. These are all broad classification of the novel drug delivery systems. The ideal properties of the novel drug delivery systems. It should be inert, biocompatible, mechanically strong and comfortable for the patient, capable of achieving high drug loading, readily processable, safe from accidental release, that is bumping effect, and simple to administer and remove, easy to fabricate and sterilize, free of leachable impurities. The terminology of novel drug delivery systems. So first of all, before developing novel drug delivery system, we should know about the difference between sustained release and controlled release. What is sustained release? Any dosage form that provide medication over an extended time period, that is called as a sustained release. It will extend for some time period. But controlled release denotes that the system is able to provide some actual therapeutic control, whether this be of temporal nature or spatial nature or both. So controlled release, it will release the drug for longer period of time, but sustained release, it will delay the release. If you see the plasma concentration versus time profile, so if you see the conventional release, so you are giving the dose, So then after some times, immediately you need to give one more dose of the drug. So like that, dose frequency is more in this system. But if you see sustained release, so this curve is sustained release curve. So it will release the drug for longer period of time, extended period of time. But if you see the control release, so it will control release. In this control release, so it is prolonged. So it is maintained, the concentration of the drug is maintained within the therapeutic window for longer period of time. This is the advantage of the control release systems. So that's why we need to develop the control release systems. The design of the control drug delivery systems. So before designing any drug delivery systems, before selecting any drug, so first of all we need to consider some factors. So first factor is biopharmaceutical characteristics of the drug, that is the molecular weight of the drug. So if you are giving transdermal delivery systems, the molecular weight should be low. So if it is having the more molecular weight, it is very difficult to permeate from the skin. So that is the biopharmaceutical characteristics, aqueous solubility, partition coefficient, drug pKa, ionization of the drug, route of administration, drug stability. So pharmacokinetics of the drug. So absorption, distribution, elimination, half-life, rate of metabolism. That is come under pharmacokinetics properties of the drug. Pharmacodynamic properties, therapeutic range, therapeutic index, plasma concentration response relationship. So these are the pharmacodynamic parameters to be known before designing any control delivery systems. So classification of the novel drug delivery systems, types of the novel drug delivery systems. So we have 4 classifications, that is the rate pre-programmed drug delivery systems, so feedback regulatory drug delivery systems, activated modulated drug delivery systems, and site targeting drug delivery systems. So first of all, so rate pre-programmed drug delivery systems. So name itself indicating that the rate is pre-programmed. So initially it is fixed to release the drug at certain rate of release. So in this system, the drug reservoir is present in the delivery system. So we are having the drug here. So the rate controlling surface. This rate controlling surface, it will control the release of the drug. So rate of release of the drug. So how much time it should be released, that is controlled by the rate controlling surface. So if you see activated modulated systems. In this activated modulated systems, so we have drug reservoir, we have the drug and rate controlling surface. So it is also having rate controlling surface, but so we need energy sensor. Externally some energy should be given to the system to release the drug. So that is example osmotic pressure, so vapor pressure, hydrodynamic pressure, so sonophoresis, electrophoresis, so this one. So feedback regulatory systems. In this feedback regulatory systems, so we have drug reservoir, rate controlling surface, we have the drug, but biochemical response or energy sensor is required. So biochemical response, so we need biochemical sensors to activate the delivery systems. So next final one is site targeting drug delivery systems. So here in site targeting, so we have the drug and drug reservoir, site targeting molecule and rate controlling surface, the biochemical responsive energy source. So in this site targeting system, the surface is coated with or liganded with some antibodies, monoclonal antibodies that will direct the drug to the particular organ. So that is the target site. So directly it is the drug is releasing at the particular organ, like cancerous cells, or sometimes it is crossing the blood brain barrier, or sometimes it is targeting to the eye, sometimes it is targeting to the colon. So this is the targeting of the drug delivery. So briefly we are classifying into the 4 categories, that is rate pre-programmed systems, feedback regulatory systems, activated modulated systems, and site targeting drug delivery. These are the 4 classifications. So first of all, rate pre-programmed systems, the release of the drug at predetermined rate. So here we have the drug, so we have drug reservoir, so we are having drug controlling surface. So it is rate of release is initially it is fixed. So again further it is classified into the polymer membrane permeation control systems, polymer matrix diffusion control systems. So these are the 2 classification of the rate pre-programmed systems. In this one, so first of all, polymer membrane permeation systems. In this polymer membrane systems, so we have the drug reservoir that is encapsulated into the membrane. From the reservoir, the drug is slowly diffused into the membrane. So here the factor is the membrane. So membrane, it will control the release of the drug. So the thickness of the membrane, so also it will control the release, the porosity of the membrane. So here in polymer membrane permeation control systems, the membrane play active role in the release of the drug. So second one is polymer matrix diffusion. So here we are mixing the drug with the matrix, polymer matrix. So the drug is slowly released into the matrix. From the matrix again it will release into the aqueous diffusion layer or systemic, so that is site of the administration. So here the membrane is controlling, so that's why it is called as a polymer membrane permeation control delivery systems. Here the matrix of the polymer is controlling the release of the drug. So these both two are come under the rate pre-programmed systems. The polymer membrane permeation control delivery systems, so that is hollow systems containing an inner core surrounded in a water soluble membrane. Polymer can applied by the coating or micro encapsulation. So here the drug reservoir is there. Here, so we are having the membrane. The membrane is slowly irritable. So now, so the examples of the polymer membrane permeation control delivery systems, so that is so NORPLANT. So this is the brand name, PROGESTASERT and AccuSERT. So these are the different types of the delivery systems that is come under the polymer membrane permeation control delivery systems. So now we will discuss about the individual products. So the rate of drug release is defined by, so what is the rate of release from the rate pre-programmed systems? So that is membrane permeation systems, that is Q by T. So Q is the quantity of the drug per time, that is called as the rate of release, that is equal to K M by R, K A by M, D M and divided by K M by R, D M HD plus K A divided by M, DH HM into CR. So we are having so many parameters here. So if you see individually parameters, the K is the partition coefficient of the drug molecules into the membrane. So surrounding membrane into the Km by R. So that is reservoir, from the reservoir into the membrane of the system. So after that Ka by M, that is the partition coefficient of the drug from the membrane into the aqueous surrounding layer, diffusion layer. So Dm, these are the diffusion coefficients into the membrane and rate controlling membrane and aqueous diffusion layer. So Hd, so this is H is the thickness of the membrane, hm, Hd is the thickness of the aqueous diffusion layer. So means the thickness of the membrane also it will control the release of the drug. Rate of release is depends upon the partition coefficient of the drug into the membrane, into the aqueous diffusion layer and diffusivity, at the same time thickness of the membrane. So whatever the membrane you are using to cast your delivery system. So at the same time, so the aqueous diffusion layer, the surrounding layer of the drug delivery system. So the thickness of the surrounding aqueous diffusion layer also it will control the release of the drug. So this is CR, this is the loading dose, the concentration of the drug present in the delivery system. So this is the rate of release. So that is the Q by T, that is having, we are having the these many parameters. So by altering these parameters, you can control the release. You can pre-program the release of the drug. So first example is NORPLANT. NORPLANT is a form of birth control. So this NORPLANT is the implant, subdermal implant. The originally NORPLANT consists of set of 6 small tubes, that is 2.4 mm to into 34 mm silicone capsules. So these are all 6 units are silicone capsules, each filled with the 36 mg of the levonorgestrel. So each capsule filled with the 36 mg of the levonorgestrel. So a progesterone used in many birth control pills. Instead of using pills, if you use this NORPLANT, so it will take care of the birth control. So implanted subdermal in the upper arm and effective for 5 years. 30 micrograms per daily. It will release 30 micrograms of the drug by daily. It will effective for 5 years. This NORPLANT, so you can see here, so this NORPLANT, so the 6 flexible capsules filled with hormones inserted under the skin on the inside of the woman's arm. So you can see this one, so how it is implanted. So what it does, the hormonal method prevents ovulation and thickens cervical mucus. How well it works? NORPLANT is approximately 99% effective. We have two types of the NORPLANTs, so that is generation 1 and generation 2. In generation 1, drug crystals alone capsulated in silicon mediated grade tubing, both sides sealed with the silicon mediated adhesive. So we have 6 units. So in generation 1 we have the 6 units that is implanted under the skin. In generation 2, so the drug solid dispersion, so here we are using the solid dispersion encapsulated in the silicon mediated grade tubing, both ends sealed with silicon mediated adhesive. So here in generation 2 we have the 2 units only. In generation 1 we have 6 units. In generation 2 of the NORPLANT we have the 2 units. Main advantages: implants provide continuous protection up to 5 years. So no need to take every day the pills for birth control. Inserts may be easily removed. Whenever you want to remove, you can remove the insert. So only single decision is made by the user about what method to use, nothing to do daily or at the time of intercourse. Once it is implanted, it will take care of 5 years. Whenever you want to remove the implants, you can remove. So that is easy method. So there is no side effects also. If you take oral dose of the levonorgestrel, so you will be having severe adverse effects. But NORPLANT, so it will overcome all the problems. So next example is PROGESTASERT. So this is the brand name, IUD, intrauterine contraceptive devices. It is also used in the contraception. Okay. Progesterone crystals. So here we are using the progesterone crystals, 38 mg dispersed in a silicon mediated fluid and encapsulated in vertical limb of the T-shaped ethyl vinyl acetate non-porous membrane. So this is the ethyl vinyl acetate membrane. So in this one, so we are incorporating the progesterone. So then this device is T-shaped. So we are having the limb. So this is the T-shaped limb. So that will be easily accommodated into the uterus. So this is inserted into the uterus. That's why it is called as a intrauterine devices. Once it is placed in the uterus, the IUD is approved for the birth control for up to 5 years. The daily 65 micrograms of the drug is released into the uterus to control the birth. Next example is AccuSert. So that is also membrane permeation control systems. So here the polymer membrane is controlling the release of the drug. The developed by the Alza Corporation. So it is oval shaped flexible ocular insert. So it will release 20 to 40 mg per hour for 7 days in glaucoma condition. So in the glaucoma we are using the pilocarpine. So this AccuSert, so that consists of pilocarpine. So it will take care of 7 days. The consists non-irritable. So it is non-irritable device. Once it is completed, then you should remove this one. What are the different parts present in the AccuSert? So drug reservoir, the material is pilocarpine. Carrier material, we are mixing this pilocarpine along with the alginic acid. This is acting as a carrier material. Rate control membrane. So we are having the 2 rate control membranes here, up one end and one is down. So this is the ethyl vinyl acetate copolymer. So this is the membrane of the EVA copolymer. So this will control. So the drug is diffused into the membrane. From the membrane, slowly it is released into the eye. The delivery portal copolymer membrane. So from the membrane, the drug is released. So we are having the annual ring to accommodate your ocular insert. This is white color ring you are seeing, this one. So this is called as an annual ring. So within this ring you are incorporating your AccuSert. So impregnated with titanium dioxide for visibility. So we are using the titanium dioxide inside the ring. So for easy visibility problem, to overcome the visibility problems. So this is about the AccuSert. So it is readily available in the market. So next example is membrane control system is TRANSDERM-NITRO. So this is also brand name. So it is manufactured by the Novartis. So we are having the transdermal. This is the transdermal system. So here you can see the membrane. This is the membrane. So from this membrane, the drug reservoir, the drug is slowly released into the membrane. From the membrane, the transderm nitro, it is stick onto the surface. So from the membrane, the drug is slowly released into the systemic circulation through the skin. A tan colored backing layer. So this is the tan colored backing layer. So it will prevent the permeation of the drugs. So upward direction. The drug reservoir containing the nitroglycerin. Nitroglycerin is the active ingredient, the drug we are using here. It's an ethylene vinyl acetate copolymer that is permeable to nitroglycerin. So this is the membrane. So here we are using the membrane as a ethylene vinyl acetate. From this membrane, from the reservoir, the drug is diffused into the membrane. From the membrane, it will go into the skin. The layer of the hypoallergic silicone adhesive. We are using the adhesive to stick the delivery system to the skin surface. It should be non-irritating, it should be easily removed. So we are having the different types of the transderm nitro, so that is manufactured by Novartis. The transderm nitro 0.5 mg per centimeter square per day for anginal attack. So angina pectoris, to treat the angina pectoris. So we are having this one, transderm nitro 0.2 mg per hour. It will release the 0.2 mg per hour. So transderm nitro, this is a one more system, it is releasing 0.5 mg per hour. So it is 0.4 mg per hour. So these are the different types of the system that is directly stick onto the surface of the skin to prevent the anginal attack. So it is the transdermal system. From the skin, the drug is released into the systemic circulation to control the angina pectoris. Next example is ESTRADERM. So this is also membrane control system. The continuous delivery for twice weekly application. Two systems are available to provide nominal in vivo delivery of 0.05 or 0.1 mg of the estradiol per day via skin. Each corresponding system having an active surface area of 10 or 20 centimeter square containing 4 or 8 mg of the estradiol USP and 0.3 or 0.6 ml of the alcohol USP respectively. So this is also ESTRADERM. So this is also membrane control system, estradiol. So it is released from the membrane. From the membrane it will go into the systemic circulation. So this is about the polymer membrane control systems. Now, so rate pre-programmed systems. So class B, that is a polymer matrix. Already I told. So this is the matrix systems. We are having the polymer matrix. From the matrix, at 0 time there is no release. Once it is exposed to the body fluids, then the drug is slowly diffused into the matrix. From the matrix it will go into the aqueous diffusion layer. From the aqueous diffusion layer it will release at the particular site. So we are having the matrix, polymer matrix. The soluble drugs, slowly dissolving matrix. Here we are having the slowly dissolving matrix. The rate of release of the drug, Q by T, that is rate of release, that is explained by the equation, that is 2ACR dP whole to the power of 1 by 2. So Q by T is the amount of the drug in the matrix system. So if you are using the matrix, what is the rate of release? So that is depends upon the dP, diffusion coefficient of the drug in the released medium. So CR is the solubility of the drug in a released medium. So A is the concentration of the drug present in the matrix per unit volume. So this is the equation. From this equation you can control the release of the drug. So that is predetermined. So that's why it is called as a rate pre-programmed system. So both membrane and matrix systems are rate pre-programmed systems. The example for the rate pre-programmed system, matrix system. So that is first one is NITRO-DER. So this is the nitroglycerin transdermal patch. So here it is using nitroglycerin using for the anginal attack. So step 1, so water soluble polymer we are using glycerin and PVA heated polyvinyl alcohol. So nitroglycerin and lactose added to above solution and molded on impermeable metallic plastic laminate. You will get the NITRO-DER. This is the transdermal patch. So it will release the drugs 0.5 mg of the nitroglycerin so per day. So it will control the anginal attack. The next one is matrix system is Frondol tape. So Frondol tape, it is developed by the Yamanouchi-Japan, this company. So this is the Frondol tape, the brand name of this Japanese company. The active ingredient is isosorbate dinitrate. So this is a vasodilator, coronary vasodilator, isosorbate dinitrate. It consists of 40 mg of the isosorbate dinitrate. So it is used for the used as a vasodilator in angina. The drug directly dispensed in a pressure sensitive polymer medicated fluid. No need of other adhesive. So this is a self adhesive device. So for transdermal application, once you apply small pressure, it will stick. It's on own. So the example is polyisobutylene, that is a isosorbate dinitrate. So it will also used in the anginal attack. The next one is COMPUDOSE subdermal implant. So this is implant system, that is a matrix system. This is also matrix system, but is used in the states. Estradiol 25.7 mg implant. So this is the implant. So this is the matrix system. 200 to 400 days. So it will take care of 200 to 400 days birth control in the. It will promote the growth promotion in the states. Estradiol. So we are using the estradiol. 200 means it will take care for 200 days. For 400 means 400 days. So it consists of 25.7 mg of the estradiol. So next matrix system is Syncro C. It is fabricated by dispensing the drug reservoir, which is a suspension of the norgestamate in the aqueous solution of PEG, polyethylene glycol 400, in viscous mixture of the silicone elastomer. So this is the Syncro C. This is also implant. Now we have discussed about the rate pre-programmed systems. So next one is feedback regulatory drug delivery system. Second category of the novel drug delivery systems, so that is feedback regulatory drug delivery systems. In this group, the release of the drug molecules from the delivery system is activated by triggering agent. Already I told. So we are having the rate controlling surface, but we need to have some triggering agent. The rate of drug release is controlled by the concentration of the triggering agent. They further classified as bioerosion regulated drug delivery systems, bioresponsive drug delivery systems, and self-regulating drug delivery systems. So first one is bioerosion regulated delivery systems. Bioerosion means biologically the polymer, whatever the polymer present in the delivery system, that will be eroded by using the triggering agent. So biological triggering agent. The system consisted of drug dispersed bioerodible matrix fabricated polyvinyl methyl ether ester, which is coated with the layer of the immobilized urease. So here, so this is the delivery system. We are using hydrocortisone mixed with the polymer. The polymer is polyvinyl methyl ether ester. So the drug is mixed with the polymer. This is the bioerodible polymer. But the surface of the delivery system is coated with the immobilized urease. This is the enzyme, urease enzyme. In a solution with a near neutral pH, the polymer only erodes very slowly. There is no erosion of the polymer, very slow erosion of the bioerodible polymer. But in the presence of urea in the body, so whenever it is exposed to the urea, so we have urease on the surface of the delivery system. This urease react with urea. So there is a chemical reaction between this enzyme, urease and urea, then it will release the ammonia. Once the ammonia is releases, obviously the ammonia is the base, it will increase the pH of the system. So surrounding pH is increased. Once the pH is increased, the polymer, the polyvinyl methyl ether ester polymer, it is having the sensitivity to the higher pH. So at the higher pH, the polymer is breakdown, so it will be eroded. Biologically it is eroded. Why we are calling this bioerodible? Because so biological triggering agent is urea here. So once the urea is attacking your delivery system, so the urea is reacting with the urea and it will release the ammonia. So then ammonia, it will raise the pH. Once the pH is raised, so obviously the bioerodible polymer, it will release the hydrocortisone, that is a drug from the delivery system. So that's why it is called as a bioerosion regulated systems. The triggering biological agent is urea here. Next one is bioresponsible regulatory drug delivery system. Bioresponse. So what is the biological response? So here also same smart delivery systems. So we are having the insulin. So release of the insulin. So no need to take any insulin by using the needle here. So once you take this bioresponsible insulin device, the reservoir contained a device enclosed by the bioresponsible polymer membrane. So this is the membrane, polymer membrane, bioresponsible polymer membrane. The permeability is controlled by the concentration of the biochemical agent. So that is the glucose. So if you see the bioresponsible membrane, so inside the bioresponsible membrane, so we have the glucose oxidase, the enzyme. Glucose oxidase is the enzyme is immobilized inside the membrane. Once the concentration of the glucose in diabetes, once the concentration of the glucose is increases in the blood, so the glucose will sensitize the membrane of the delivery systems. Then the insulin is released. The glucose triggered insulin drug delivery system. This is called as a glucose triggered insulin delivery system. So I will brief about this delivery system, how it will work. So in this one you can see this is the bioresponsible membrane, I told already. So I explained. So the glucose oxidase is present inside this one. So we have the ammonia group NR2. So in this system the insulin reservoir is encapsulated within the hydrogel membrane having the NR2 groups. In alkaline solution, the NR2 are neutral and membrane is unswollen and impermeable to the insulin. So it naturally at normal pH, so the NR2 group is there intact. So there is no permeability of the insulin from the delivery system into the membrane. Once glucose, the concentration, so once the blood glucose levels are increased, so obviously they penetrate into the bioresponsible membrane. So this bioresponsible membrane already consists of glucose oxidase. There is a reaction between the glucose, the blood glucose with glucose oxidase. So because of this reaction, so what happened? So the gluconic acid is formed. So the acid is generated. Glucose reacted with glucose oxidase. So oxidation of the glucose, it will increase the pH. It will decrease the pH. Glucose oxidase, gluconic acid. Gluconic acid is the acid. It is producing the protons. These protons reacting with the NR2 groups. So here the NR2 groups. So there is a hydrogen protons are given to the NR2 groups. Glucose penetrated into the membrane. It oxidizes enzymatically by the glucose oxidase entrapped in the membrane to form a gluconic acid. The NR2 groups is protonated. So there is a protonation of the NR2 group to form NR2H plus, and the hydrogel is membrane is then become swollen, it will permeable, so it will become very loose. So then the insulin is permeable through the membrane. So here, so the bioresponse membrane, so once the glucose concentration is increased in the blood, so glucose oxidase is present in the membrane, so glucose react with glucose oxidase to produce the gluconic acid. Once the gluconic acid is formed, so it will produce the protons. The NR2 groups, intact NR2 groups, so that will take the protons, and the once it will take the protons, NR2 groups, it will become more flexible, and then so it is more permeable to the insulin. The insulin is released. The triggering agent is here is insulin concentration. So it is insulin concentration, glucose concentration. Once it is increases, the insulin is released from the delivery system. So this is called as a bioresponsible systems. Next one is third one is self-regulated delivery systems. This type of systems depends upon the reversible competitive binding mechanism of the active end to regulate the release of the drug. The drug reservoir is drug complex encapsulated within the semi-permeable polymer membrane. The release of the drug from the delivery system is activated by the membrane permeation of the biological agent from the tissue in the system is located. So example I can give this self-regulated system. So there is a competitive binding. So this is the cancavalin A. So this is the molecule. So we are having the membrane. The cancavalin is enclosed into the membrane. This cancavalin, so that is the glycosylated insulin is attached here. So 4 glycosylated insulins in a cancavalin A. Once the glucose concentration in the blood is increases, glucose will permeate into the membrane and it will go and it will be competitively bind to the cancavalin molecule. So the glycosylated insulin. So that glycosylated insulin is competitively bind to the, so this glucose is competitively bind to the cancavalin molecules to release the insulin molecules. So this is the insulin molecule, that is the glucose is going inside, it is attaching competitively, it will bind to the insulin and glucose is replaced here. Then the insulin is released into the systemic circulation. So this is the cancavalent molecules glucopenetration into the system. It activates the release of the glucosylated insulin from the complex for controlled delivery out of the system. Whenever the concentration of the glucose is increases, because of the competitive binding, the insulin is going out. So glucose is into the system and insulin is out. So this is about the self-regulatory drug delivery systems. So this is two types is completed now. So activation modulated drug delivery systems. Activation modulated drug delivery systems already told. So the energy, external energy is required for this one. So we can classify this activated modulated system. So activation is done by physical means. So you are applying the osmotic pressure, hydrodynamic pressure, vapor pressure, mechanically activated, magnetically activated. Magnetic field is you are applying to release the drug, and sonophoresis, that sound waves you are applying, iontophoresis, and hydration activated systems. So that is a physical means. So physical factors are involved. Next, chemical means. So pH activated systems. So we are using the pH sensitive polymers. So that is the, so example, colon targeting, only the polymer, whatever the coating we are giving to the system, the polymer is released only at the higher pH values, not in the stomach, only in the intestine. So that is ion activated systems, ion exchange release, ion exchange resins, and hydrolysis activated systems. That is the chemical mean. Next, third one is biochemical means, enzyme activated systems, so biochemical activated system. Thank you for listening this lecture. Thank you very much.